All right, so the one thing, for those of you who have started on the homework, somebody pointed out to me that you can only upload one file for the first question. So if you'll do the paired read one file, and I added that to the question, so you'll see it. So if you've already started on it and you were confused, and we'll see more about the trimming today, and that might make more sense. I'll point it out when we get there. We should produce the file you need today, and then all you need to do is run FastQC and upload it for the homework. Okay. I'm going to go ahead. We have a lot to cover today, so I'm going to go ahead and get started on the slides. How do I get to the little thing? Okay, so we're going to talk a little bit about short read mapping. So you may be thinking, okay, well, we already covered how alignment works. We spent a whole class period on BLAST and how to align reads. But it turns out there's a lot of trade-offs to be made depending on the type of data that you have and what you want to accomplish. So there's a big trade-off between speed and accuracy, and the faster it is, usually you're able to detect, it's not necessarily less accurate, you have less sensitivity. We talked a little bit about global versus local. It's another decision you have to make. So different types of alignment objectives lead to different categories of aligners. So what you'll find is that bioinformatics is just like there's a million different types of read aligners, and they all do different things. And today we're specifically going to cover short read mappers. So BLAST is a lot faster than the original algorithms we talked about. So we talked about Smith-Waterman as another example that gives you an absolute guaranteed optimal solution. BLAST is a heuristic. It gives you an approximate solution based on some clever ways to speed up mapping. But BLAST is still way, way too slow for the amount of data produced by a next generation sequencing technology. We would still be waiting years and years and years to map single runs. But we have a specific thing that can kind of save us. Since resequencing, you're often looking at reads that are very, very similar to your reference, like 90% plus. We can go ahead and create an algorithm specifically targeted at reads that are very, very, very similar to the reference genome. And by leveraging, the constraint that we're only going to look for very exact matches, the software can get much, much faster. This will also generally utilize a global strategy instead of a local strategy, again, to speed things up. Several tens of millions of reads can be mapped per CPU hour. So this was a huge improvement over BLAST. But in general, for most short read mappers, about 95% identity or greater are all they can handle. So if you move to a different species and you're trying to map back to a reference genome that's quite diverged, you may have very low success rates. I tried this with an oak genome, and when I took DNA reads from one oak species and tried to map them to another oak species, only like a small percentage mapped. And Narula has done this as well for his project. Turns out the genic regions are quite conserved. The non-genic regions are way too diverged, even within the same genus, for a short read mapper to work. Short read mappers are also, like with BLAST, you want generally all the hits that are above a certain cutoff. Short read mappers are all about finding the best hit or the set of equivalently best hits. That's just sort of their role when you have next generation sequencing data. And that's because the point is to find where they originated in the reference genome. You don't necessarily care what other homologous regions might be. And so you're just going to discard those. So some reads, in the majority of the cases, you're going to be happy when your reads map uniquely to your reference genome. You know where they came from. You think you found the correct region. However, genomes are complicated. They have repetitive elements. So some of your reads are going to map to multiple locations. This can be pretty difficult to figure out what to do with those for many downstream applications. For RNA-seq, for example, if your reads map to 2 different genes, how are you going to count that? What is your strategy to figure out which one they actually represent? For a SNP, or if you're looking at variation, where does the variation actually exist in your genome? If a read maps equivalently well to 2 different places, you don't necessarily know. So a couple strategies to deal with multiply mapped reads. So the first one is just to throw them away. And this is honestly probably the most common thing
that people do with next generation sequencing data, if they have a reference genome, is just toss them and say, I can't make heads or tails of this. It's not worth my time. However, this does introduce bias, depending on the type of question that you're trying to answer, and it's ignoring real genomic regions that may be biologically important. We're not going to discuss that much for DNA-seq or for variant calling, but for RNA-seq, we'll discuss that a good bit more, because there are strategies for trying to use that data in RNA-seq. Next generation sequencers will try to use a couple of clever ways to try to find what is the best alignment. The first one is that they're very sensitive to quality values. We learned about those crazy, the FASTQ format, where you have a letter or a number or a symbol representing the quality. The most recent next generation sequencing aligners will actually use those qualities and say, if there's a mismatch at a low quality base, I'm going to penalize that less than a mismatch at a high quality base. So pretty subtle, but a nice way to try to find the absolute best match. And of course, paired in information is very helpful. If one of your pairs can be uniquely mapped, you can deduce where you think the unique location is for its mate pair based on that fragment length. So let's say you have a read, it matches the beginning of chromosome 1 and the middle of chromosome 2. Its pair matches 100 bases downstream at the beginning of chromosome 1 and has no match to chromosome 2. So you go ahead and just assume that both reads belong on chromosome 1 because they originate from that same DNA fragment. So that's an example of how paired in read information is really helpful for getting unique mapping for your reads. Does that make sense? Okay. So as with all software, there are many decisions for the end user. How many mismatches are you going to allow for a read to be considered mapped? So you can, to some extent, play with, I really only want things that are very, very, very similar versus I want to try to look at things that are a little more diverged. If you have an incomplete or a low quality reference, this may be more important to you. There are software packages you can pick that are more friendly to this. How many matches do you want reported? A lot of these software packages will give you the option. Do you want to know only best matches? Do you want to know the whole set of equivalently best matches? Do you want to know everything above a certain threshold? And of course, as we've already seen with Linux, the parameters and the names of the parameters and the flags are all going to be completely different between each and every software package. So you will have to read the documentation and really think about what you want to do, and maybe read papers that other people have done the same things and see what parameter they used. But it's important to basically say, is it doing what you think it's doing? Tons of popular software packages. These are some that I see a lot. BWA and HiSat2 are probably the ones I see the most. Most of them have the most boring names, which always makes me sad because there's this opportunity as scientists, we don't always get to name things. You can name things something fun and nobody ever does. But I came across this, which is called Velocimapper, and I thought it was great. I've never tried it because it costs money, and I don't pay money for software. It's just like against my philosophy. But I thought it was a really clever name and a nice logo. So if any of you guys create bioinformatics software, I demand that you give it a cool name and a fun logo. Yeah, so all of these are good. Some of them are older, some of them are newer. Stampy is a particularly good one if you're trying to go across a large amount of divergence and heterozygosity. The rest are not as friendly to that. There's this great little website that shows all of the short read mappers that have come out, and I think these are actually only the open source ones. And below this great chart is a table with all of their features. But there were a few years from like 2008 to about 2015 where you couldn't open a bioinformatics journal without seeing a new short read mapper. It just got to be completely out of control. So now that's sort of slowing down. I feel like people are like, okay, this problem has been pretty well solved, moving on to other bioinformatic challenges. But basically there's just tons and tons of different packages that you can choose, and lots of them have iterations. So there's Bowtie, and then there was Bowtie 2, and then Bowtie 2 kind of got overtaken by HiSat, which got overtaken by HiSat 2. So some of the same groups will keep publishing. So try to figure out if you're on the most recent version and the most recent iteration of their software. So how do you choose? All bioinformatics software, this is not just read mappers. Look for good documentation.
Try to ask probing questions of the documentation, and if it's like a one-page PDF that makes no sense, stay away from it. That's not going to do you any good. Memory efficient is nice, especially depending on what kind of computational resources you have. If you're reading a paper and it says something about, oh, this is particularly memory efficient, or we compared it to these other algorithms and it's just as accurate but more memory efficient, that's going to be helpful. Google and see if you can find a mailing list or a help forum. That generally means that somebody will help you if you run into trouble, and also that they're actively maintaining it, so that when bugs are found or as new operating systems come out, that they're actually maintaining the code and making sure that it's still going to work. If you find something from 2007 and the code hasn't changed since 2009, you know, likelihood of it working these days is low. Yeah, the most popular ones, like I said, are pretty similar in mapping rate and time. BWA and HiSat2. HiSat2 is a little faster. BWA is generally beloved by many in the community. It's like really the most accurate. And I ran that past our like official genotyping guy, and he said BWA is still the one that everyone loves. But both of them are quite good. Neither one is very good if you're trying to go across a lot of heterozygosity or a lot of differences. So that I would say Stampy. I think there's another one, but I'd have to look it up. So if that's particularly interesting to you, find me after class and we'll look up what those are. Similar to BLAST, most mappers have indexing strategies. So you're not just going to go in and run your mapping. You're going to first index the reference genome. So instead of having a database with lots of proteins or genes in it, we're going to have the whole reference genome and maybe one sequence, like a microbe, and maybe many chromosomes, and maybe tens of thousands of scaffolds. But whatever it is, there's usually a step where you index it. And this is very much like I explained with BLAST. The point is to make it into a format that's really suitable for very, very fast searching. Like an index, like back when we had physical paper books and you turned to the back and you looked up the index and it said, oh, this word is found on pages 13, 47, and 102. That makes it much faster instead of you trying to read the whole book every time you need to find a new concept. This is how Google works. When you type something into Google, it does not go search the whole internet just for you. It is keeping an index of all the keywords and where they all live all over the internet. So that's why you have to do it. Burrows-Wheeler aligner is what BWA stands for. And that's what we're going to be using today. It has 3 algorithms. We will be using BWA MEM, which is by far the most common. A caveat here, individual chromosomes cannot be longer than 2 gigs. This is almost never a problem unless you work on conifers or some other plant that has crazy, crazy, crazy long chromosomes. But worth noting, because it will totally fail if you have a single sequence that is longer than 2 gigs. I know output in the SAM format, which is a totally standard mapping format, what everybody prefers and wants to use. So that's another good thing that should probably be on the list of things if you're looking at a mapper. Make sure it outputs in a standard format. SAM would be the format you care about. So BWA MEM will work for any sequences greater than 70 base pairs up to 1 megabase. So if you have super short reads, like 25 MERS or 50 MERS, like really short reads from Illumina, you won't want to use this. But as long as your reads are at least 70 base pairs, you can use them. If you have 100 base pair reads, this is tolerant up to about 2% error. If you have 200 base pair reads, it's tolerant up to about 3% error. So as the reads get longer, it tolerates a little more error. This is not a problem for Illumina. Illumina is generally less than 1%. So unless you just are concerned that you had like a really noisy run, like this shouldn't present a problem. One of the great things about it, not all aligners are smart enough to split reads and say, I actually think there has been an insertion in your reference and in your sample as comparison to the reference, and that your read is actually half of it's over here and half of it's over here. So BWA actually has that ability to try to identify if a read has been split, which is really nice. That becomes particularly important for RNA-seq because introns. But we'll get there. We'll talk about that when we get to RNA-seq. And it does report multiple alignment locations, which does not always work well with other software. If you do not want it to do that, you can research the --M flag, and that will help you to make it report only a single alignment location. Yes, so it will report multiple alignments that it thinks are all equivalently good. So it's not going to report this alignment, this location is really great, and this one's kind of like not that great. It's going to say, oh, I see 3 locations, and I think they're all equivalently good. So I'm going to give them all to you. I think with the dash M flag, and I haven't looked at it in a while, but I think it randomly picks one of those to report back to you. So that's something you want to think about. Do you want, you know, the randomness is kind of a concern, especially if you're, depending on what you're doing, that could be of concern. If I were variant mapping, I would probably just throw away multi-mapped reads. So all of those things are flags that you could research. So I want to do just a little bit with SAM, BAM, and CRAM format. Formats are like half this class. It's either like debugging, like trying to install software that won't install, or like figuring out file formats. That's like the bread and butter of bioinformatics. So another new file format, SAM, stands for the Sequence Alignment Map Format. This is the tab delimited plain text version of this format. And it's basically, it is meant to store large nucleotide sequence alignment. So once you run a short read mapper, you have all these locations where all these reads go against a reference genome. How do you store that in some kind of manageable way that you can put it into different software packages? So it includes alignment of every single individual read. It can handle gaps, variants, and structural variants. It handles pairing of reads. It handles more than one alignment location in the genome. It stores the quality values for both the mapping and for the individual nucleotides, and it stores information about duplication. So it stores a lot. The strengths are that it's very flexible. You can shove a lot of different types of information into these files. And it's useful for operations on very large sequences. And it has really good documentation, which is great. And it has its own software package called SAM Tools that will allow you to manipulate and do all kinds of things with these files. So here's the basic structure. The header at the top of the file, all of the lines are going to begin with an @ symbol. And that's how you know you're in this kind of header metadata area of the file. Below that, each and every read gets its own single line. And you'll know that it's a read because it won't start with an @ symbol. So the header contains information about what was mapped, what it was mapped to, and how. It always includes the version information for the format itself. So it'll say, this is a SAM file version 1.3, or this is a SAM file version 1.4. So as the SAM file specification changes, if you receive an old SAM file from a collaborator, you can figure out what version it is. Whether or not and how the file is sorted. We'll see there are a couple different ways to sort these files. Information about the reference sequence. So what were these reads mapped to? It generally stores the software version, any flags that were used for the software, so all sorts of different kind of metadata at the top. It's really great if you're trying to write the methods paper, the methods section of your paper, and you can't figure out exactly how something was mapped, look in the header file, the header part of your SAM file, and it'll also tell you like what software, what version, what flags, and that's great for writing your methods. Of course, you will be writing that down anyway, because you're all very responsible scientists who would never not write that down. But just in case, you know, a collaborator sent you a SAM file and you had to figure that out, that's the place to look. So here's an example. It's pretty cryptic. I generally have to go look up every single line in the specification. But this, for example, is the version. So this is version 1.5 of the SAM format. And the sort order is coordinate. It's sorted by coordinate. It can also be sorted by name. Below that, it has a line for each reference sequence. So if you map to a reference genome that has 10,000 scaffolds, each and every one of those scaffolds will have its own unique little header line. And it says, I know about a reference sequence. The name of this reference sequence is this, and the length of that reference sequence is 45 or 10,000 or 1 million or whatever it is. So that's kind of nice to know. It gives you some information about both what was mapped to and how it mapped. But again, all of these little 2-letter codes are standardized, and usually you just have to go look them up in the specification. So when you get out of the header, then you get down to the actual like tab delimited part of the SAM file. It has 12 or more tab delimited fields. So if you suck this into an Excel spreadsheet, this would be at least 12 columns. You can't have more. The first column is always the query template, which means basically your read name. The second column is a flag. I will talk a bit. The next slide is about what's in the flag. It's complicated, but it's a number and it has lots of meaning. The next column is what part of the reference, so like chromosome 1, chromosome 8. The next is the one base leftmost position on the chromosome, so starting from base pair 1, base pair 2, base pair 3. Where's the start of this read mapping? It has a mapping quality, which is essentially the same as the Phred scaled quality we've already seen. So there's a quality estimate of how likely is this to be correct. Cigar strings, we also have another slide on, but that tells you exactly how the read mapped. And then within the same line, it goes ahead and if there's a mate, so if there's a mate to your read, it tells you the name of that mate, where it mapped, this is easy for display, so you can show them as pairs. So they'll both be in there. It's like having their own line, but they'll also have the mate information in the same line that they have, if that makes sense. Like they both have their own line, and they include the information from their mate. It tries to infer the template length. So if you have these pairs, it tells you how far apart they are, or if they map to different chromosomes, that maybe there's some kind of structural variation here. More quality information, and then a bunch of optional fields that we'll talk about in a minute too. So tons of information. So this is an example of a single line from a SAM file, and we're gonna unpack this field by field. So the first thing is just the sequence name. This should match up to the sequence in your FASTQ file. It should look exactly the same as your FASTQ file. Field 2 is this flag. So the flag is taking a bunch of different numbers, 1 through 2048, and you kind of have to think about it like a checkbox. Like if I checked off that the template has multiple segments and they're all properly aligned, and their PCR optical duplicate, I would take 1, 2, and 1024 and add them up and put that in this field. Which seems like a very complicated way. This is a very computer science-y binary way to do things. It doesn't actually work. It's basically using ones and zeros to store information, but the way you visualize it when you open a SAM file are these base 2 numbers. So if you get whatever number, like 83, and you can deduce that that is 64 plus 16 plus 2 plus 1, and then you look it up in the table, and you realize that means the read is paired, it's mapped as a proper pair, it's mapped to the reverse strand, and it's the first in the pair. There is a calculator online where you just type in your number and it tells you all the flags that it corresponds to, because this is actually kind of a pain in the butt to figure out as a human. Super great for computers, not so great if you're a person. It's not often that I'm actually like trying to figure out a single read, but if you ever need to, you can. And here's the explain flags.html. You just type in the little number and it like tells you all the little like ones and zeros and what they mean. The next field is your reference. So what reference sequence it mapped to. This should also correspond directly to the FASTA file of your reference. The position where the first matching base maps. The mapping quality, which is essentially the same thing as Phred. So a map quality of 30 would be 99.9% correct. A map quality of 0 would be the probability that it's correct is like nothing. It's absolutely not correct. If you get the value 255, that means that the mapping quality is just missing. So,
I'm sorry, could you go back to the original SAM file and show where that 60 where you are? Are you?
Okay, so here is the query read. Here is the flag number that you would try to figure out what the heck that is with the little calculator. This is the reference, which wraps because it's kind of long. And then,
so the 60 is kind of in the middle of that second line.
The 60?
The field.
So the 83, okay, the 32.95725.
Field 5, so that's what I'm trying to find.
The right here. Okay. So there's a tab there. It just didn't render on the slide very well. Should probably try to put it in like a table.
So that's field 4.
2, 1, 2, 3, 4. Yeah, 60.
And so 5 is that 60, just that 60 there.
Yes.
Okay.
Mapping quality. Yes. Sorry, I have not. It would be better in a table. But when I just copied and pasted it from a file, and I think the tabs, you know how it's like in Word, depending on how your offsets are. Like sometimes your tabs are different lengths. Yeah. But the slides should match up back to the original example. So yeah, if the slide says 60, then it's 60. So yes, this would be like 0.00001% probability of wrong. So I think this is actually one of the best quality. I think this might be the best quality value you can have. So up to 60, and then if you get 255, that's a not available. The quality value is missing, and that can be if you have a mapping software that isn't very good at supporting the SAM format. It may just not fill that field out, which is hopefully not something that you're going to see. The cigar string. So this is actually an extremely clever way to store alignment information. And you guys remember we saw in BLAST, and we'd have like the reference sequence and the query sequence and dashes where there were gaps and like little lines if they were the same, and like trying to figure out how to store all that is very bulky. So they came up with this sort of shorthand way to store alignment information. And so you walk from the left part of the alignment over toward the right. And so if you see a bunch of matching bases, you would say, okay, I saw 6 matching bases. I'll call that 6M. And then you have an insertion in the reference. You might say 1I. And then you hopefully you don't see a lot of like clipping and padding because those get really complicated. And like this one, 8S28M. That means 8 nucleotides are clipped off of the front and then 28 match. But it's basically a way to have like very, very few characters describe how many bases were matching, and then there was insertion, and then some more matching bases, and then there was a deletion, and then we clip the end. You can store all of that information in like 10 characters. Super, super short. Again, as a human being, you're probably not going to look at these very often, but in case you ever have to, if they're in there. There's software to visualize these alignments much more intuitively. If you're looking at a SAM file and you're like, what the heck is this thing? It's called a cigar string. That's what it is. So here's another example where it starts at position 5. We have 3 matches, so 3M, 1 insertion, because there's no reference base to pair up with this A. So that's 1I. 3 more matches, so that's 3M. A deletion, so it's always in terms of the read, not in terms of the reference. So in terms of the read, there's a missing base, so that's 1D, and then 5 more matches, so it'll be 5M. You would see this, and you'd be like, wow, what is that thing? But it translates to exactly how the alignment was structured. Does that sort of make sense? Okay. Oh, I guess I was gonna walk through it a little more. Next year. Okay, so the next field, not the last field, the 7th field, the reference sequence for the next read in the template. So you can look and say, okay, did my reads map to the same reference, like the same chromosome? For forward read, this would be the reverse read. For the reverse read, this is where the forward read maps. If you just see an equal sign, that means they're the same. And again, this is just to save space. So if your reference name is 30 characters long, you can just slam an equals in there and save yourself 29 characters. The position where the next read maps. So again, this makes it easy to do on-the-fly calculations about whether or not your paired reads are actually mapping in a reasonably good relative location to each other. Finally, if your reads are paired, it's going to do this. If they're not paired, all of these will just be blank. But it's going to try to give you the template length, so including read 1 and read 2 and the gap in between them. So the whole DNA template that was sequenced from the leftmost base to the rightmost base, and it will make it negative if it's reverse complemented to the reference. Then it has the sequence of the read and the quality of the read. So pretty much everything that was important from the FASTQ file gets restored into the SAM file. And then this optional more information. And this is very much like the header, where you have just tons and tons of really cryptic 2-letter codes that software packages tend to use. Some use some codes, some use other codes. It's not always super standardized, but you can go in and look all of these up in the specification. For the most part, we're just not going to worry about these because there's a ton of them. They differ for every read mapping software. But variant calling software will often use these to determine important information. I think I have some examples here. Edit distance to the reference, template independent mapping quality, number of best hits. The number of best hits is nice to know because that's how you figure out if you have multiple best hits for sequence in your file. If you want to store suboptimal hits, normally you don't, but if you did, that would be reported that this was a suboptimal hit. Number of mismatches, number of gap openings and extensions, and just all sorts of random information that might get put in this like last field. Okay, so this is the overall SAM. You have the header at the top with the @ symbol, and then each and every read gets its own individual line below that. So if you get a SAM file and you get a file and you don't know what it is, if it looks like this, it's a SAM file. Okay, the sister format to the SAM file is called BAM, and it is the binary version of SAM. And basically, it is a type of zipped file. It is a fancy blocked new zip format. So it's a little bit bigger than gzipping, but it's easier for a computer to access the middle of the file. So yeah, a computational, like a piece of software can load the file, look up information in the middle without loading everything before and after. So the way it's compressed is good for file access. It is up to 75% smaller in size than the SAM file, which is nice. It is not readable by people. If you do a head or a tail on one of those files, it's going to look like this. This is how you know you've got a binary file in your hand. So this is computer readable only. But there's a software SAM tools that's going to allow you to convert back and forth between human readable and machine readable. It's generally nice to store things in BAM format because they won't take up as much room on your file system, and you can always convert it back to human readable format if you need to. But as we've seen, like that's not that human readable. Like, I mean, this is a stretch to call this human readable. So for the most part, you're not going to want to look at this. I mean, maybe if you have like a question about like one area of your reference, you really want to dig into it, you might look at it. For the most part, store things in BAM and let the software do all the work reading them. CRAM is the newest format, which I think they thought they were being very clever with the word CRAM. It's fun. It hasn't really taken off. There's some software packages that use it, some don't. It's an even smaller and more efficient version of BAM. It's still pretty rare, but if you see it, I just wanted you to be aware that it's out there. And there's like different types of CRAM, which I don't think really helps with people taking it up because that made it a little needlessly complicated. But if you run into it, SAM tools will convert between CRAM, BAM, and SAM. So if you end up with one of these files, SAM tools will also handle it. All right, any questions about short read mapping or SAM files?
What exactly is compressing a file? What's the computer doing?
It's getting rid of all that space. Like we need lots of like spaces and like, it's getting rid of space, and it's taking letters that were stored as like 8 bits, and if it doesn't need all 8 bits, it'll like smush it down to 4 bits. And it's just an algorithm, and I don't actually know a ton about how it works, but it's able to just pull out all that excessive space and get rid of all the unnecessary bits and shrink it way down, but in a way that it's still recoverable back to human readable.
There's a lot of strange symbols there.
Yeah, okay, so that's because like to a computer, the letter A is like 000001, and letter B is like 00010, and so it's just strings of ones and zeros, and they're delimited by a certain number of bits. And when it compresses it, all of those bits, like lots of the bits get removed, and so when it tries to turn it back into text, all of those letters look random, if that makes sense. Like all your ones and zeros have like shrunk, and so now all the symbols, it's not really storing the symbols, that is a text editor attempting to interpret your file as text when it is no longer text. It isn't a totally different thing. So it's just like gobbledygook, right? And that's why it looks like that. Yeah. Good question though. And there's a lot of work on like how to make things super small. As you've heard from like bioinformatics lately, especially, is all about like, how do we make all this data much smaller? I read a paper about how we're at that tipping point where it is almost cheaper to resequence samples than it is to store the data. That the data is getting so big and the sequencing costs are getting so small that we should biobank the samples and throw all the data away because it's more expensive to try to store and back it up. If your samples are precious, that may not work, but I mean, if you have like plenty of sample left. Okay, so we're gonna go on to the practical now. Okay, so the first thing we should talk about is that I realized we had a really big error in our last lesson, and I didn't realize it until yesterday. When we downloaded, or when I downloaded the FASTQ dump files from NCBI, they're totally wrong. They're like this, it's the same setup. The read 1 and read 2 are duplicated in both the _1 and the _2 file. I've never seen this before. I have no idea what that means. I did a bunch of research on Sunday trying to figure out how that happened, and I still don't really know. So I re-downloaded everything and redid the lesson from last week to make it right. And I'll tell you, and I almost thought we should like go through and look at the error, because it took me a while to like go all the way back and figure out what had happened. But when you try to map those reads, the mapper says there's no paired in information. I kept saying, no, 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 these are paired. Like I kept trying to force the mapper to treat them as paired. And the mapper kept saying, I can't identify pairs. So then I started looking at the FASTQ files, and that's when I realized that like, if you do head, and I was just really quickly doing head and saying, oh, they look like FASTQ, moving on. But it turns out they're 100% identical. It's not a read 1, read 2 situation. And I have never seen that before with FASTQ dump. Normally it's a pretty accurate program. But I believe that everyone who used FASTQ dump, and certainly everyone who copied the files from me, has this problem. So if you've already done the homework, which might just be you, I don't know if anybody else got ambitious enough to do the homework, feel free to turn those in. I'll check both. It'll be fine. But we're gonna recopy everything so we can start with like a real clean, actual paired in data set in the correct format. So I guess I have this a little bit out of order. So yeah, let's log in. And then I talked to, I emailed the ACF help desk after last class period and said, okay, we're up to half the class being able to log in. And they said, okay, split the class in half and have half try the class project and half try the UTK condo project. So if your last name begins with A through N, copy this QSub command. If your last name begins with O through Z, that should split us almost perfectly in half. Copy this QSub command. The only thing that's different is one of them is using our class project allocation and one is using like the global UTK allocation. Okay, so while we all wait on those, let's go back up here. So I took the correct files and I reran FastQC, and now we can go look at them. So they're linked off of this DNA practical page. And I put links. You can go do a lot more research about what the each individual plot means. In this case, it's giving you a breakdown of the quality value for each position. So base 1, base 2, base 3 as an average over your entire data set. And this is only for read 1. We see that we have very high quality. So on average, our quality scores are very high. It doesn't like this. So this is kind of interesting. I see this in almost every single Illumina run we ever get back. And I put a link to some information about this. Random hexamer priming, which is what they sell you when they sell you a kit for generating next generation sequences, is these hexamer priming sites are totally random and they're going to introduce no bias in your reads. But it turns out that's just like absolutely not true. So there's a whole breakdown that somebody did of like, yes, every person on earth is seeing this and FastQC is kicking them all out as not random, and it's not. But according to this person who wrote this, said it doesn't seem to have a problem. And I would agree that when we look at our reads mapped to a reference genome, it won't look like our reads are piling up very specifically in certain locations. There is enough different hexamers being used that it looks pretty random, but it is not truly random. And that's why you see this, and that's why you should not worry about it. I used to trim it off, but since reading that, I just leave it and don't worry about it, and I don't think it impacts anything. GC content. This is peaking at a location that I find to be pretty normal. I guess I should probably go look up the GC content of E. coli and make sure that the peak is right where it should be. But in general, as long as it's evenly distributed, I don't worry about it too much. Even though it gave us a yellow isolation point, like this would not worry me at all. There are no ends. All of our sequences are still in 245 to 249 range, so they should all be exactly 250 or 249. This is usually a good place to look for problems. If you run into a DNA or an RNA bottleneck and you have a lot of PCR priming that has to go on and you end up amplifying the same fragment over and over and over and over again, you'll see that pop up as duplicates. In this case, we see that most of our reads do not look like duplicates. They all have a duplication. Like this is the percent of our seqs that have 1x, they're unique, is very high. And there's a few where we have 2 or 3, and that could be random, like you just randomly sample the same fragment from the genome. And there's a little bump down here, and that could be repetitive elements. But if you see that your sequence duplication is peaking way down further down on this x-axis, then you might want to get concerned. This very much depends on the type of sequencing you're doing. So, you know, if you're doing 16S, where you're going to be preferentially amplifying the same region over and over and over, this is a much different meaning for that, right? You don't expect it to be random. You weren't looking for random. No overrepresented sequences, and this automatically checks for the most common adapters, and it didn't find any adapters. So all of that is good. And again, this random hexamer primer, it's going to tell you what hexamers worked really well in your library. But for the most part, the rest of this doesn't really, it's pretty good, to be honest. This is generally what we see as far as overrepresented K-mers. If you have a lot of adapter, it will generally show up here as well as adapters out front or at the end. Yeah, those are the non-random hexamers at the beginning. Yeah. Okay, so interesting things to note. So yeah, let me see. Actually, let's look at the very top of that one again. So here, all the way to the very end looks quite good, right? It looks like, you know, we've got a few bases we might want to trim on a few of our reads. Let's go to read 2. Much lower quality. This is not always the case, but pretty frequently read 2 is just in way worse shape than read 1. And I think that's because the whole process of getting the reads to bend over and then do the polymerase on the other end, things go wrong. For whatever reason, read 2 usually, this is pretty drastic. Like I don't see this very often. So it's a good example. But be aware that you may end up with lots of reads that are, read 1 is 250 bases, but read 2 needs to be trimmed down to like 100 bases or 50 bases. And there's still a lot of useful information in there. It's just read 2s are often just less good. So if you see that, don't be surprised. And I clicked on per base in content. Some of our bases have ins, and I don't, that is not usual for an Illumina run. So you'll just get used to looking at these as you do more and more sequence data. But it doesn't really, it just, the first base is often in. I don't know why. And this is not something I've ever gotten data that looks like this, but you know, I would have flagged it as like, that's weird, why is that? I might have asked the sequencing center, like, was there something that went wrong with the instrument on that first base? Because it's only peaking the in at base 1. So yeah, I gave you links to go examine whatever your FastQC report says for your sequence data. There's lots of information about what it might mean if you go to the FastQC website. Okay, yes.
For the second sequence, would you mind going over the per sequence quality score chart and the difference between that one and
The per base sequence quality?
per sequence quality, yeah.
This one?
No, keep going. There, that.
Yeah, this is weird too. And I think it's just reflective of, you know, do they have the, like, I'd wonder if a whole sector of a slide was bad and then it caused like a whole sector had like lower quality values and the rest of the slide was okay. Because again, this is not something I've really ever seen in a sequencing run that we've gotten back in real life. It usually peaks and it has a much more normal distribution. But sometimes like the sequencing facility will send you like the whole lane or the whole slide, like one whole sector can go bad. And that would be my suspicion here, is that something went wrong with that. Like the camera wasn't quite focused or something was weird. Because it looks like a lot of things are really high quality, but you had something went wrong with some portion of the reads. And what caused that? I would call the sequencing facility if I saw this and be like, hey, what do you guys think? Have you ever seen this? It's not typical. Normally you see a normal distribution. The second read 2 might be lower, but it's usually also normal. So yeah. It's kind of like if you ever need to argue with the sequencing facility that like, I gave you a lot of money, you need to fix this, like these graphs can be very helpful. Yeah. And you can always go on Seek Answers and like post them and be like, what do you guys think? Because the people on Seek Answers, it's a great website. People are very helpful. Yeah. Okay, so how many people had their jobs launched? Mine is still not launched. No. Oh, okay. Well, that's good. Okay, how many people who are in the latter half of the alphabet had their jobs launched? Okay. Okay, so few, not that many. Okay, for those of us who are in the latter half of the alphabet and it didn't work, let's try the first one. Oh no, I was hoping that they were all. Apparently we crashed a whole node last period, which I did not feel guilty about at all. So they're trying to figure out something for us so that we can all log in. Yeah, this is unfortunate. It's kind of a more computationally intensive lesson than the last one for sure. What else can we talk about while we wait? Oh, this is the directory structure that we have started on, and when I get you to copy the corrected files, I've named them E. coli fixed. And so continuing to practice having like good directory structure, keeping all of our data organized, this is what your everything should look like. And then we'll be adding analysis folders as we go over this class period and the next 2. So I'll try to post a diagram for each class period so that you can follow along. But just keep this kind of structure in your mind as what it should look like. Okay, so for those of you where this has worked, CD into your home directory in the class project directory. And if you do an ls at that point, you should see E_coli. Next thing you're gonna do is copy, and let me know if the permissions don't work. the E. coli fixed from my directory into your directory. And you'll use the copy-R so you can get all of the contents of that folder. Okay, so who still does not have a job launched? Oh man. All right, well, I guess we'll use login node. Don't know what to do. All right. So if I go to you should now have an E. coli fixed. And if you ls the E. coli fixed, you should have analysis and raw data. And if you look at raw data, you should have the same files. This time they're actually correct. Your FASTQ files are actually correct. So let's actually go look at those real quick so that you'll catch this problem if you ever see it. I'm just going to get one sequence. So that's Shasta-N4. And then do the second one. So let's see if I can make this. So the way we know these are paired is that we have this unique name and 1 out of 1. And then in the other file, it has that same unique name and 1, 2. So it's just forward and reverse. They're labeled 1 and 2. So originally what we had was the exact same sequence with the exact same name. And you should notice these are different, right? Like this one starts with ACAT and this one starts with TTAC. So these are actually 2 different sequences, a forward and a reverse. If you do a head on your _1 and a head on your _2 and they are perfectly identical, then something is wrong. Again, I've never actually seen that happen before. So hopefully you won't either, but something to look out for now that we know it exists. Okay, let me get this out of the way. Okay, so I'm going to back up and go back into the analysis folder. And at this point, you should have one FastQC and that's it. And so what we're going to do is create a directory called to-trimomatic, and cd into it. So if you do module avail, it's going to tell you that Trimomatic is installed on the system. I couldn't get it to work, so I just reinstalled it in our class directory, and that just means we have to use the full path to get it to work. Maybe one of you can figure out how to get the ACF version to work, but it's a Java file, so installation is really just copying it from the internet and putting it in an accessible location. So if you go to the course webpage, there's a huge long command that we are going to use. And I'm going to go ahead and hit enter and let this start running while we examine what's going on here. Let me drag this back over. If you copy this whole thing, and then hopefully it should start giving you feedback and say it's doing stuff. So while that's running, let's break this whole giant command down. Again, this is something where you just have to read the documentation to figure out the order. So you put, it's a Java program, so you actually have to tell it to run it with Java. You give it this jar file is the name of the software. The very first parameter that you put in is your forward read. The second parameter is your reverse read. And after that, these 4 files are all the names of your output files. So you're actually creating, this is what you want your output to be named. You want your trimmed sequences that are still paired to go in this file. Your trimmed unpaired in this file for 1 for the forwards, trimmed paired and trimmed unpaired for the reverses. So what that means when you have it, you start out with this 3 pair. They both pass, yay. Trim 1 pair, trim 2 pair. But if you're, let's say read 2, terrible quality, it gets tossed. It's gonna get rid of it entirely. That remaining forward read is going to go into unpaired but trimmed. So that's how you end up with 4 files. You can have both of your reads survive, or just your forward read survive, or just your reverse read survives. For practical purposes, most people don't use these unpaired reads. If you have enough data, you probably don't care about them. You just want to use your paired reads. But you have to tell the software where to put them. It's just a requirement for Trim-O-Matic. If you have single in reads, this is all a good bit easier to set up because it's pretty much just this is my input and this is my output, and that's it. But because they're paired and Trim-O-Matic really wants to try to save as much data as it can for you, it's going to give you back those unpaired but trimmed reads. From there, you have to give it a location for your adapter sequence. You do Illumina clip and colon, and then this whole long path to your adapter sequence. So that whole, that's a path to a file in your, somewhere that you have access to. In this case, I use TrueSeq3. I think that's actually right. If your sequencing company sends you the adapters, you would shove them into a FASTA file and put the full location here. And I think these are the kind of the recommended ways. You can adjust how stringent it is at adapter trimming. Those are the default. I think they're fine. And then at the very end, you have some more little directives. We didn't actually need these because our quality looked okay, but just as an example, I said trim off the first 3 bases from every read, no matter what the quality is. Trim off the last 3 bases of every read, no matter what the quality is. So if you knew you had like something, like you'd looked at your FastQC and saw that your first few bases were always really bad, you could just go ahead and hard trim those. And then I have a sliding window. And the way the sliding window works is this is saying, go through the sequence from beginning to end, and for the first 4 bases, if the average quality value is above 15, keep it. Go to the next 4 bases. If the quality value is above 15 on average, keep it. Go to the next 4 bases, and so it slides along, looking, and as soon as your quality value dips below 15, it trims the rest of the read. So that gives you, if you have like 3 really good bases with like 1 bad base in the middle, that's probably still gonna survive. But once you get toward the end of the sequence where all of your quality is going down, it's gonna go ahead and trim it. And then your minimum length. So if you've gone through all your other trimming and you have less than 36 bases left, toss the whole thing. So lots of parameters, lots of flexibility, a little bit complicated, but that's how it works. Do you guys have any questions? There's a big manual. Every piece of software you ever run, you should go try to read the manual and understand it, because there's generally more options than you could ever dream of using. But some of them you may actually really care about. Okay. So at this point, green stickies, who has a .trim.paired, a .trim.unpaired for read 1 and read 2? Yay. Okay, most people do, that's great. So the homework, the first homework question is to take, and I just edited it today, take this .trim.paired read 1 file, run FastQC, and upload it to the homework. So this involves a couple of skills. You have to remember how to run FastQC. You have to figure out how to get it to your laptop with secure copy or FTP, and then you have to upload it to Canvas. So homework question 1. So at this point, you may want to look at what did the trimming do? How much data did I lose? And what are some ways we can do that? So one thing we can do is, let's look at the number of reads. So here we're using some of our Linux command line skills. I want to count the number of lines in my raw FastQ files, which are up 2 directories, back down into raw data. If I put a star, I'll get 1 and 2. And then I want to compare that to my number of lines for my trimmed. So I'm just going to get the FastQ files in my current directory. So this is, of course, if you wanted the actual number of reads, you would have to divide these by 4, because we know there are 4 lines for every read in a FastQ file. we're just eyeballing it, we can say, oh, you know, I went from 8.2 million reads originally to 7.76 million. So I didn't lose a ton of data, right? I lost about 500,000 reads, but that's not totally unexpected. These two should always be the exact same number, and these two should always be the exact same number. If you have a 1 and a 2 file that are paired, because each pair should have a member, a 1 member and a 2 member, right? A forward and a reverse. So they should be the same. These are going to be different because we preferentially have thrown away a lot of read 2s because they were lower quality. So we end up with these like orphaned read 1s. They're all on their own. Their partner has been discarded. It's very sad. And then these few reverse reads that their forward partner was discarded. So these are kind of our orphans. So these guys won't necessarily have the same number. These two always should. We can also look at the total file size. And we'll see that our file size has gone down pretty significantly from, and this is interesting too. Our file sizes are a lot different from each other. So even though there's the same number of reads, the read 2 is much smaller. That's because it's been trimmed more heavily. So it has significantly fewer sequences. So once you run FastQC, you'll get the number of reads and average length. You'll be able to do all that. This is kind of the quick and dirty way to start looking at how many reads and how much data you lost from your trimming. Okay, so we are going to follow, if you go to the SAMtools webpage, okay, let's just do what I would normally do, which is Google SAMtools. So samtools.sourceforge.net always pops up first, and you can click on it, but where you're actually going to end up is down here. But we'll click on it to just show you because it's still the first hit. It basically says at the top, for the newest releases, go to this other website. So this is where you actually want to be. It's called HTSLib. And this was a big effort to write a big computer library so that software packages could interact with SAM files and BAM files. And they rewrote SAMtools and this other package called BCF tools that we'll see later to use this library. So there's like this big sea change. Like there was old, like SAMtools was its own thing and BCF tools was its own thing. And then they did this huge rewrite. This is where you want to be. These are the better packages. These are the up-to-date packages. The other thing that this has is all of the manuals and specifications. So you can click on SAMtools for the software package. You can go down for the file formats and learn all about these file formats. So really good resource. And finally, it has workflows. So these are pretty good workflows. This is not the absolute best variant calling workflow, but it's pretty quick, which is important because we don't have forever. And it walks you through very nicely exactly what you want to do to call your variants. So this is essentially what we're going to follow for calling variants. If you ever need to do it on your own, it's a good place to start. Okay. Okay, so we are in Trimmomatic underneath our analysis folder. I'm going to back up one folder. I can now see FastQC and Trimmomatic. The third thing we're going to do is mapping, so I'm going to create a new folder for that. And I'm going to CD into that folder. BWA is an ACF module. I'm just doing module load BWA. It is not the most recent version. It's a few months, it's probably like 6 months out of date. If you ever run across that on the ACF and you want them to update it, you can email the help desk and say, hey guys, there's a new version of the software. Could you please update it? Because I prepared this lesson like less than 24 hours ago. That didn't get done. But it is worth checking. Like, is the module on the ACF actually the most recent version of the software? Some of them are very out of date. So be sure to email the help desk if you need a more recent version. Okay, so we're going to build the index.
Whenever you load whatever other program
It shouldn't. If there's a conflict between the two, it'll just tell you there's a conflict, I can't load that. But as long as they aren't conflicting, they won't. Like, so how could a conflict arise, right? So there's Python 2 and Python 3. And so if you load one software package that uses Python 3, but you already had something loaded that required Python 2, it would be like, I can't have two versions of Python loaded right now. This is impossible. But for the most part, if they don't have conflicting dependencies, it's just going to keep them all loaded all at once. Yeah. Yeah. If you put this in a shell script, especially if you launch it, we'll learn how to queue sub things. You might have to put this in the shell script to get it to pick up, to load the software. And every time you log back in. So once you log out, it gets rid of all of that when you log back in. But as long as you're in one session and you've stayed logged in the whole time, the software is still loaded. Yeah. Okay. So tab complete is your best friend. BWA index. I'm going to go back, 2 directories, raw data. I just type GCF tab. It'll get me up to here. I type a lowercase g, and it'll get me the genomic. And then I think I actually have to type like .f to get to the FNA. Each time you tab complete, it'll help you get a little closer and save you a lot of typing. If anyone's having problems, raise your hand. Nicole can come around and help you. Okay, so now we have another kind of long command that I'm going to copy and paste, and then we'll go through what it's doing. So here's the whole command, and I'm just going to let that run in the background while we talk about it. Okay, so BWA, because it comes with 3 different types of software, your first parameter that you give it is always which one of the 3 things do you want to use. In our case, we're going to use MIM, BWA MIM. This is really handy. We would not have necessarily needed this for our example because we only have 1 E. coli strain. We're comparing 1 strain back to the like quintessential E. coli reference sequence. But let's say you had 50 strains and you had 50 files, and later you wanted to call variants and you wanted to be able to determine alleles and trace them back to your strains and actually do some biology. You would want to name them appropriately. So this is basically saying in my SAM file, in that little header, when it says read group at RG, I want to make sure that the sample has the ID that matches up with what I think the ID should be, the sample name. So in this case, I put the ID of the sequences in NCBI and the sample, the biosample accession, and the library. So you could have something really complicated going on where I had this strain, I sequenced it once on an Illumina HiSeq, I didn't get enough data, so then I sequenced it again on an Illumina NovaSeq, and I did, so there were like 2 library preps and 2 instruments, and have all this data going back to 1 strain. So this just gives you the flexibility to specify an ID for the file, the actual biosample name, so the strain or whatever, and a library number, so if you had multiple libraries. So again, we wouldn't have needed this if you only have 1 sample. But if you have lots of samples and very complicated experimental design, this is super helpful. -T specifies the number of threads you're going to use. This scales pretty well. So using 2 threads is going to make it almost twice as fast. Using 4 threads is going to make it almost 4 times as fast. Since we're each getting a single CPU, for those of us who had Q subs, it actually worked. 2 threads should be able to run on 1 CPU, so that's not going to overstress the system. But if you had a computer with 10 cores, you could do 20 threads and have it run really fast. Again, this is just looking at the documentation to know the order of these parameters. You always give it the reference genome, the forward read, then the reverse read, and then you say shove all of my results into a SAM file named this. So these little like slashes at the end of the lines, I think I mentioned this, but just to mention again, that's basically saying ignore the new line. I'm not ready to enter, I'm not ready to submit this yet. Wait for more information. So each new line just lets you, I mean, we could have this on one giant long line, but you would have to scroll forever to see it. So the little slashes at the end of the line just help us to break a single long command line up into a more readable version. So yeah, if you only have one read, like a single-ended read, you only specify that. You don't have to specify both.
And there's lots of other parameters. Here's the manual in the upper right. And this is going to give you like kind of the general patterns for how you run BWA. And then just like your manual file, well, first you go to mem. Like the parameters are different depending on what your first parameter is. So as long as you've typed BWA and then mem, all of these parameters can be used. Let me make that bigger. So we talked about seed length with BLAST. That is similar here too. So you can play with the specificity. Longer seeds are going to find it's going to be faster but expect more exact reads. Shorter seeds are going to allow for more mismatches but be much, much, much slower. If you see a read that maps more than twice or more than 10 times in the genome, you might want to discard it. Yeah, most of these I don't really mess with too much. Yeah. So you might want to use this. I've heard this is helpful for variant calling. I didn't put it in the lesson, but I think some protocols that I've seen online say to always use the -M. Yep. So if you have any questions about what you can do with BWA mem. Oh, and this is how I figured out that our reads were all messed up, because it was saying skip orientation FF, skip orientation RR, skip orientation FR, skip orientation RF. And I was like, but that's every possible orientation. You can't skip them all. There has to be like, I expected the FRs to work. And I kept rerunning it and I couldn't figure out what was going on. And then 30 minutes later, that's when I figured out our reads were all messed up. So sometimes, even though it's just a warning and not an error, like it didn't spit it out and say, I can't do this. It was just giving me these like really unexpected messages. And I was like, huh, that doesn't make sense. So sometimes even if it doesn't like die and say, no, this is not working, you still need to think about some of these messages. I mean, a lot of them are kind of just updates, like how many reads have I processed? But some of them are important. Like if I hadn't paid attention to those skipped orientation ones. And I kind of think when I taught this class in 2016, I think we might have used the wrong reads the entire time and I never noticed, which makes me feel bad. Huh?
Why is it skipping?
Because we don't have forward or reverse. We have forward reverse. I don't know. Like I had never really thought about it before, but it was saying I can't find forward. Okay, that's not normal. I can't find reverse. Okay, that's not normal. And then it said I can't find forward reverse, which is normal. Like I think I mentioned this with paired ends and mate pairs. They're called like innies and outies. So the paired ends are innies, so you have forward and reverse, and the mate pairs are outies, so you have reverse and forward. I don't know what kind of library prep gives you forwards or reverses. I've never seen those, so I always expect to see that. But yeah, I like forward reverse is what I expected. So when it said it was skipping that orientation, I was like, I don't think that's right. And that's when I.
So then what is it doing, since we're waiting anyway, what is it kind of doing at each?
It's taking a hunk of sequences from the file and it's going to the index and it splits the little sequences up into seeds. And then it goes and looks them up in the index. And then it says, okay, so I've got these 5 base pairs in this sequence and we've got these 5 base pairs in the reference. Let's go see if we can turn that into an alignment. Then it goes over and it tries to work upstream and downstream and see if it can align the read. And if the read aligns well enough, it passes a quality threshold, it gets shoved into your SAM file. And if it doesn't, then it just gets, well, still get shoved in your SAM file, but it'll get shoved in your SAM file as like did not map. So that's what it's saying, is it's like taking these chunks of reads and turns it into the little seeds, and then it goes, compares it to the index, and then it goes to the reference, and anywhere the seed exists in the reference, it tries to extend it. So it's surprisingly similar to BLAST. It's just the underlying algorithm is just different, and it's faster, but it requires a lot of sequence identity to work.
So is it reporting every time it analyzes the read, or how often is it reporting?
No, I think it's just doing like, actually, I don't know if it's triggered by the number of reads it's processed or by the amount of time that's passed. See if we can figure it out. So that was 82,608, 82,704. So it was like another 106 reads. Yeah, these might be like standard hunks. Oh, it might be, and this is probably what it is, at 200 or 20 million bases. It takes a hunk of 20 million bases. It looks like it's always right at 20 million bases. So it looks like it's taking a hunk of 20 million bases, and then it reports back on how long it's been. Then it goes get another hunk, and then it, yeah. So I think this takes about 4 minutes. So it might be a little while. I tried it on HiSat, and HiSat took less than a minute. So I really like HiSat. It's at least not as good. So while we're waiting, I can show you another way to make your code really reproducible. So by the way, you can have multiple terminals open. Like a lot of people don't realize this. Like often you just want to have, you're waiting on one thing, you want to keep doing stuff. You can just open another terminal. Oh, and I haven't filled out the sheet yet, but I'm gonna teach you guys how to do this like SSH ACF thing, and you don't have to type your username and everything. I'm gonna put that on the homepage for the course. Okay, so you can see my SAM file is already being built as it's running, but don't start trying to use it or mess with it until it's done. But one of the things that's nice to do is once you have your command figured out, paste it into a file, and I'll also often just call it command.sh, and you can open it with Nano. And then I might paste in my whole command. And so this is gonna be nice because I can always come back to this directory and see what I had in here. And if I accidentally delete the SAM file, I can turn this into an executable file and just rerun it. So save the buffer, write it out. So let's see. This is not executable right now. You can see that there's no execute permissions, but I can do U plus X command. And now if I ran something like this, don't hit enter because you already have it running on the side. But I could do something like that and rerun the command without having to type it back in. And if I came back months from now, I would still have a record of exactly what I ran and be able to rerun it with no effort to like try to figure out what I had done or what order I put the parameters in. Like it would all be there. So I recommend doing this for each and every analysis folder you have. Like store your commands in a file in that folder. All right. And feel free to have lots and lots of terminals open and be doing stuff. You can do lots of things in parallel. Don't be afraid to have lots of terminals. Okay, so we have our SAM file. Let's look at the top, the head of the file. So this one only has 3 header lines. We have a line that tells us the name of our reference and the fact that our reference is 4.6 million bases long. It gives us the ID number and the sample number and the library name that we specified. So you remember up here we had this -R flag, and we specified that we wanted the ID and the sample and the library to look like this. It followed the directions and made all of those things happen here in the SAM file. And then this is just saying that, okay, this file was generated with BWA. This is the version of BWA that was used, and this is the entire full command line. So if you had not managed to store that in a file or had lost it, SAM files will try to tell you exactly what command was run to produce this file. So that's actually really convenient. And then we have our crazy long SAM file format, just like we would expect. So we've got our sequence name, our flag that we could go back and deconstruct. If we only have one reference, it's E. coli, so it's just got like one genome. But if we had chromosomes, this would say like chromosome 1 or chromosome whatever. Where it mapped, very high mapping quality, 251 matching bases. The reverse read was mapped to the same location, to the same reference. This would be the leftmost position. So from 1,451,920 bases for the forward read to right downstream for the reverse read. And thinks the total sequence DNA fragment was about 420 bases. We've got our sequence, then we've got our quality values, and then we've got a bunch of flags that I would totally have to go look up to know what they mean. But again, like it's nice when you get a file to just be able to do a head and a tail and determine like, does this look like what the format that it's labeled as and the format that I expect? That's actually a really good skill to have. How many people have a SAM file? Okay, most people, great. Okay, so now that we have a SAM file, we can start using SAM tools. So SAM tools is this great software package meant specifically to just do everything under the sun with SAM files. I ended up installing SAM tools, I think also in our software directory. So let's see, module, avail SAM tools. They have 1.3 is the ACF version. The version online is up to 1.9. I thought it was a big enough gap that we should go ahead and try to use the more recent version. 1.3 is, I think that's more than a year old. So I went ahead and installed 1.9 so that we would be able to use it. But unfortunately, that means you have to type the entire path. So Lester, Haven, courses, EPP, software, SAM tools, then SAM tools. So inside of a software folder, if you're looking for the actual executables, they're sometimes in a folder called bin, which stands for binaries. So the binaries are what are executable. So if you're ever just looking for software, a bin folder is a good place to look. I am going to do the stats function. So just like BWA mem, where like your first parameter kind of define what part of the software package you're going to use. Go ahead and do stats. I'm going to use my SAM file. And I'm going to output this to a new file. Oh, this is another good use of tab complete. I didn't want to type all that stuff again, so I typed dr tab, and it filled it up. And I'm just going to back up and call it stats. So I didn't have to type all that, right? Tab complete. It's wonderful. Did that make sense? Sort of. You can copy and paste it from the course website too. I mean, I have had that complaint from this class before where people get really used to copying and pasting and then they can't figure out how to construct commands on their own. So like, don't get too attached to it. But we also move pretty fast. So if you're not a fast typer, that can hold you back in the labs. But when it's your own files, of course, you have to sit down and construct all of these commands yourself. So be aware of that. These stats files are really big. So I'm going to open it in Nano. So it gives us some header information. This file was produced by Sam Toolstats. It gives the command line. And then down here, you get total raw sequences. How many were first fragments, how many were last fragments, so forward and reverse reads. It looks like 40.8 million of our 41.3 million, or no, 4.08 million of our 4.1 million were mapped. So that's a huge percentage of our reads that were successfully mapped. Slightly fewer were mapped and paired. So sometimes the first read will be mapped and it cannot find the second read. That may be an indication of insertion or something that's structurally different about your strain as opposed to the reference strain. But the vast majority are mapped, and they mapped in a location that makes sense that they are indeed a pair. I found no duplicated reads, no QC failures. Yeah, so just a whole bunch of information. The number of bases that are mapped, the number of bases that were trimmed off is 0, the number of mismatches, the error rate. So our error rate was 0.01, so maybe about 1%, 0.001, so like 1/10 of 1%. Our average length was 241 bases. So yeah, average quality of 32, that's very high. Insert size average was 407, and your insert size standard deviation is 168. So again, this is another great metric. You can see how you're, if you're doing your own library preps or hiring someone else to do your library preps, how well is your size selection actually going? This is a great metrics for understanding that. What is my standard deviation and my size selection? Yeah, and this goes down like, if you go through the Sam Tools documentation, there's a way to take all of these charts and turn them into pretty pictures, but we won't do that. I'm going to Control X out of that. Okay, so following along with the workflow, and I don't understand exactly why this happens, but apparently BWA does some kind of funky things with the read pairings, and you need to go clean them up. So it just recommends that you run Sam Tools, and there's something called fixmate. And it's a problem with how BWA stores the mates into the SAM file, and Sam Tools has written a function specifically to fix it. So I'm gonna run that while we talk about it. So in this case, instead of stats, I'm gonna use fixmate. And as long as we're doing something with Sam Tools, I can go ahead and say, put the output in BAM format. I want to convert out of SAM and into BAM. So go through and fix the mates, but while you're doing that, turn it into a BAM file. So it's just going to compress it way down. This is the input and this is the output. So I'm going to name it. I try to name things intelligently so you can go back and figure out like, what is this file? I'm going to add underscore fixmate so that I can tell this wasn't just a straight SAM to BAM conversion. There was also like fixmate got run in there so that I can tell them apart. This might take a minute too. Actually, let's go look up what it says about fixmate, because I don't even know. Okay, so the workflow information says, because BWA can sometimes leave unusual flag information on SAM records, it is helpful when working with many tools to first clean up read pairing information and flags. And then if I go to the SAM Tools documentation, which I don't know if I still have open. we're gonna search for fixed mate. Fill in mate coordinates, eye size, and mate-related flags from a name-sorted alignment. So not a ton of information. Yeah, I don't know why. I've seen this in a lot of workflows that they recommend you do this generally almost every single time you run a mapper. So you could do the fixed mates, but I don't know why they aren't sorted correctly the first time. But this is this output format where we can determine, we can go ahead and output it in BAM or CRAM from SAM. So nice feature. You can kind of do two things with one command. Here are all the things that SAM tools can do, which if you're doing variant calling or really mapping at all, SAM tools is going to be your friend. So view is primarily to convert between the formats, sorting, indexing. So I'd encourage you, we're gonna see a bunch of these, but well, we're gonna see not gonna see a bunch. So go ahead and because SAM tools is very powerful. Okay, let me do LS/LH. So we fixed the mates, which is really just like barely tweaking one field in your SAM file. So it shouldn't really change the size that much. So this is primarily due to the SAM to BAM conversion. We went from 2.4 gigs, which is huge considering what we started with, which was not a very big read file. And we got down to 840 megs. So that is well over 50% compression. So that's the power of going from SAM to BAM, is you just save a ton of space. And the last thing we'll do before we'll quit is sort. Currently, because your FASTQ file is sorted like alphabetically or numerically, and the reads are numbered numerically, that's how it's stored in the SAM file. That's really inconvenient if you want to call variants. You want to walk down your reference from like base 1 to base 2 to base 3 to try to look at all of the reads that are aligned. So you actually want to sort all your SAM file records by where they line up in the genome. So all of your reads that begin at base pair 1 are listed. And then all the reads that begin at base pair 2 are listed. And then all the pairs. So basically we need to resort the file. And SAM tools provides a utility for this called sort. So I'm going to copy and paste that one next. So again, this is just all about reading the documentation. It's the sort function. We want to output in BAM. So we're inputting in BAM and outputting in BAM. So don't want to change format this time. I'm going to give it an output file name where I keep the fixed mate. Okay. Those of you who Q-subbed, yours should work. Those of you who didn't, or if you get this error, just make dir temp, and then we're gonna do operator SAM tools, then SAM tools. Sort -o BAM -ODR. Okay, so that's the part that's different. If you got that error. Should like that. Okay. So the reason why this matters, this actually trips people up all the time. Let's say you wanted to sort 20 files that all live in the current directory that's the same. The way SAM tools is sorting is it creates a set of temporary files, or kind of like segments. I think about it as like, if you had to sort a deck of cards, you might put like all the spades and then all the hearts and then all the clubs, and then go through and do 2, 3, 4. So you might subset it. and do your sorting in stages. So that's how SAM Tools does it. But it needs these like kind of temporary files to do that. And it wants you to specify a location for those temporary files. And the reason why this is important is because if you specify the same location for multiple sort algorithms for multiple files at the same time, they all clobber each other and your sort gets all messed up. So each library or each file, BAM file, needs its own unique directory for its temporary files. So what I had originally said was to use /temp/DRR021342. I don't know why it would already exist. I guess maybe I accidentally did this on the login node yesterday. But you should be able to access this /temp directory on a node. It's just a free-for-all. They get erased every so often. Don't put anything in /temp that you don't want to disappear immediately. It's called scratch space. And you can put anything in there and then it just goes away. So I moved to doing it in my current directory because apparently or else we're all trying to clobber each other's files, right? Like if we all use the same temp directory or the same command, it like SAM tools, my SAM tools and your SAM tools and your SAM tools would all trying to be used that directory and it would be a problem. So this temp directory thing is kind of a pain in the butt. So I just changed it to use the directory right in the folder so that it would be unique to me and each person. If you do it in your own directory, it should be unique and they should not overwrite each other. Hopefully that makes sense. But it's important to name them differently. So if you were doing 20 files and you had 20 samples, you need to name all of your temp files something different so that they don't overwrite each other. Yeah. So this will take a few minutes. So I guess we can go ahead and wrap up. If you guys want to wait, you will need it to finish prior to the next lesson. So if you have time to wait and you can say great, if not, you can kill it and just rerun it later when you have time. But I don't think we have time to do the last little visualization. So that's it. If anyone wants to stay and work on the homework, please feel free to do so. I will be here until 5.